Namaste and welcome to our poster. I am R. Shashank from the final year of BTech Bioinformatics from the Department of Bioinformatics, Shastra deemed to be University, India. Efficient degradation of plant biomass is an important goal in several applications, especially the production of bioethanol. Mixtures of biomass degrading enzymes are used in the industries for this purpose. However, the conventional glycoside hydrolase enzymes used are unable to act on crystalline polysaccharides, making the degradation process inefficient. Lytic polysaccharide monooxygenases are a family of enzymes discovered recently that act synergistically with the conventional enzymes in the lignocellulosic degradation process. Our aim through this study is to retrieve, analyze, and aggregate existing LPMO-specific data using a self-developed pipeline and represent it as a knowledge network with corresponding ontology terms. The hybrid workflow we developed for this study consists of three components. The data retrieval and curation part takes care of the retrieval and sorting of data obtained from different experimental and literature databases, including PubMed and Gene Expression Omnibus. The extracted data will be stored as indexed JSON format documents in the MongoDB database. Standard NGS data processing and identification of dysregulated genes from the stored RNA-seq datasets is performed by the second component, for which tools such as FastQC, TrimGalore, HiSat2, and DSeq2 will be used. The third component uses BioBERT, a transformer-based deep learning model for natural language processing of biomedical literature using methods such as word tokenization, sentence tokenization, and named entity recognition to generate ontology terms specific to LPMOs. Data from all the components will be pulled together finally to produce the different nodes and relationships between them represented as the knowledge network. A search facility will be implemented on the network to fetch nodes based on user queries. The first two components have been implemented as an automated pipeline in Python and R. Implementation of the third component is currently underway. Figure 2 represents the schematic figure of the knowledge network. Each node in the network represents one of the 6 categories: substrates, RNA-seq datasets from the GEO database, organisms, published literature, authors of published literature, and the LPMO encoding genes and proteins. The extracted data after curation has been segregated into one of these 6 categories. The edges connecting the nodes are directional, representing the direction in which the relationship is represented. Mapping between all the nodes except the mappings involving the gene protein node has been implemented and hosted as the LPMO knowledge base network using the Cytoscape JavaScript library at www.bioethanol.network. Figure 5a and 5b show the network obtained by searching for the query aspergillus and cellulose respectively in the knowledge base network. Mapping between the RNA-seq datasets, the fungal organisms, and the substrates is illustrated here. Each query searched against the GEO database consists of an organism name and one of the 5 carbon source names. A total of 237 datasets from 14 fungal organisms were obtained using the initial queries. Results from the differential gene expression analysis of these datasets will be used to establish the GEO study LPMO coding gene relationship in the knowledge base network. The dataset with accession GSC 51091 corresponding to the keywords Neurospora crassa and glucose was selected as the model dataset to test the knowledge base pipeline. From the list of 10 LPMO encoding genes annotated in the Neurospora crassa genome, which was obtained from the KC database, the 4 highlighted genes were differentially expressed under the experimental conditions. Co-expression network analysis of these differentially expressed genes was performed using the WGCNA package in R. Each node here represents a differentially expressed gene, while the edges represent correlation in their expression levels. The nodes are also hierarchically clustered into modules of highly co-expressed genes represented by the different colored nodes. However, differentially expressed LPMOs in the model dataset didn't show any significant correlation with other genes. Analysis of such co-expression networks constructed from other datasets might highlight novel relationships within and between the genes of interest. The networks will be stored in the Neo4j graph database and made available as part of the LPMO knowledge base. Further, LPMO-specific ontology terms generated by the NLP component will help in predicting new relationships among the nodes in the network. As part of future work, this fungal knowledge base will be expanded by including multi-omics information from bacterial species too. Thank you for listening.